Um, just a word of introduction, how this invitation came to happen. Sean and I were at SeaTac and then both on a flight to Heathrow. He was going to, as I remember, a malaria meeting in London. I was going for a vacation in Egypt. And I asked him what, in retrospect, was a very naive question. It was still sort of the height of COVID. And I asked him why it was so relatively easy to make a COVID vaccine that was effective in 1 year. And it's taken decades to try and make a malaria vaccine that's only partially effective. And he reminded me COVID was a very small virus with an easy target, and malaria was a big parasite with lots of evolutionary change, and it was a much more complex task. So when I got back here, I decided, well, that would be a very interesting thing for us to learn about, um, and hence the invitation. So happy to have you here this morning. Um, so we're going to talk about the quest to make a malaria vaccine. There's been a lot of press about this, especially in the New York Times recently, about the fact that there are now, I think, multiple vaccines, but we'll hear that story right now. Go right ahead, Sean.
Great. Thank you. Hopefully you can see my slides, and
Yeah, yeah.
please feel free to interrupt at any point. And I'm terrible at watching the chat window, so just shout it out if you're so interested. But yeah, it was great meeting you, Leonard, and I hope your trip was great. It's kind of fun how these things come about sometimes. So I have some disclosures. We've been fortunate to work with a number of startup and biotech companies that are interested in the malaria and vaccine space, Scenaria, Malarvex, and Lumen. And then I, with some other people in Seattle, started a nucleic acid vaccine company. And anyway, the slides have been reviewed, and all of this is disclosed to the UW as well. So my lab is in South Lake Union, and the department I'm in is in lab medicine and pathology. I'm a clinical pathologist, and I help run the microbiology services. I can't see everybody on the call, but some of you, we may have managed patients together talking about blood cultures and what have you. I'm getting excited if anybody has malaria on our service. So our lab is interested in making a vaccine, and we leverage mouse models, non-human primate models, and human challenge models, all here in Seattle. And then our lab also houses a CLIA-CAP certified malaria diagnostic lab that serves as a WHO reference lab. We do some testing on patients here in Seattle. We still use blood smears as the gold standard here in Seattle, but we use this diagnostic test, this molecular test, for clinical trials and for other sort of niche uses to explain unusual cases. So we talk about all that. It's not lost on this audience, the power of vaccines, but I really like this data from looking at COVID attributable deaths, hospitalizations, and infections that were averted by the US vaccine program over the first couple of years of COVID vaccines. And, you know, when we first rolled out COVID vaccines, the comments were about how amazingly protective they were. And then over time, people started to chip away at that. And, you know, vaccines are imperfect. The malaria vaccines are much more imperfect than COVID vaccines. And yet, even with these imperfect vaccines, this 3.2 million deaths averted just in the United States, just in a couple of years, is really a testament to how impressive these are. And the kind of impact that a malaria vaccine, especially a highly effective vaccine, could make over years and decades far exceeds these numbers. Up until a few years ago, there really was we couldn't claim that there was any vaccines approved for eukaryotic organisms. Plasmodium falciparum and its related plasmodia are all apicomplexan eukaryotic organisms, and everything else on this list for the last couple hundred years is either a virus or a bacteria. And so that's, of course, starting to change. And we'll spend a good chunk of today talking about the two WHO approved vaccines, RTSS and R21. We've also been making great gains against malaria using tools that for the most part have been around for some decades. Since 2000, the malaria map, whether you look at deaths or you look at these are death mortality maps. You can look at morbidity, et cetera. You could look outside Africa, but sub-Saharan Africa is the place with the highest amount of mortality due to P. falciparum. And you can see that this map is less intense from 2000 to 2019. However, COVID has definitely dealt a blow to that, as has other developments like insecticide resistance, et cetera. We'll just very briefly review the life cycle of the parasite, thinking about the way that vaccines might be used to intervene at certain bottlenecks. And the two bottlenecks that are the most important are the liver stage, where there's relatively few number of parasites. And over here in the mosquito on the left side at the gametocyte stage. But we're going to focus most of today on the liver stage, where either antibodies or later CD8 T cells can potentially eradicate all of the infected cells before the parasites emerge and start the consequential, clinically significant blood stage. In the blood stage, you really are just left with antibodies to combat the parasites because red cells lack class 1 MHC and can't be targeted by CD8s. And then later on, as you know, if you have gametocytes, those can be transmitted to mosquitoes that can go on to form sporozoites. And there's a whole other class of vaccines being developed called transmission blocking vaccines that potentially could interrupt this, this part of the life cycle and actually make a really big public health impact. But they're not like any other vaccines that you've ever given your patients, because a transmission blocking vaccine has the greater good for the public, but doesn't necessarily confer a greater good to the individual vaccinee, which is sort of an expectation of getting a vaccine if you're willing to sort of deal with some of the side effects that might happen. So we're really interested in liver stage vaccines over here on the right, where either through antibodies or CD8s, you have the potential ability to completely interrupt the life cycle. So there's a whole bunch of different vaccines that could be made, including one against the blood stage. And each of these might have different goals of a vaccine that's either perfect or imperfect. So on the left here, if you have the perfect vaccine, it's one that in the liver, at least if you have a perfect liver vaccine, it takes whatever that incoming inoculum is and it renders it null and void by the end of the liver stage, such that nothing comes out the other end and you can't develop blood stage infection. So then the infection's over for you. If you have an imperfect liver vaccine, it might reduce that incoming burden so that you start with a lower blood stage infection, and maybe that lower blood stage infection bends the curve and you suffer less severe disease as you follow this dotted line off to the right. And that's as compared to someone who's unvaccinated, the solid line that has a liver stage that starts higher and then goes on to cause symptomatic malarial illness. Similarly, if you had a blood stage vaccine, the gray dashed line here, maybe it would start to interrupt that cyclical infection of red cells and you wouldn't get as severe disease, but it wouldn't do anything in the liver stage. So you can imagine all the kind of nuances of how these might work. There are many different vaccines in the pipeline. And this is data that I just sort of cultivated about a year ago. There's certainly more vaccines in the clinicaltrials.gov pipeline since then, but this is phase 1 and phase 2-3 vaccines targeting the different stages of the parasite. You can see the majority, especially of early phase vaccines, are in the sporozoid and liver stages, and that's predominantly the kind of vaccine that's made it into phase 2 and 3 studies. There's quite a lot of work across these other stages, but particularly for transmission blocking vaccines, it's been that there's not a lot that have made it into phase 2 or 3 studies. One of the difficult things you might imagine with a transmission blocking vaccine is showing that there's a benefit to the community. This means that you might have to enroll tens of thousands of people to show that effect size. Whereas it's a little bit easier to show efficacy earlier where you have an expectation of protection in your vaccinees. So this is a table just showing many different vaccine products on the left that were all intended to somehow interrupt transmission. And you can see here they're categorized as either pre-erythrocytic. I remind you that pre-erythrocytic is a code word for liver stage or sporozoite stage, all of the stages that precede the blood stage infection. And you can see here that there's been variable numbers of these kind of participants protected. This table's a little bit old at this point. Some of these studies using products against CSP, we'll talk about CSP more, have achieved much higher rates of protection at this point if you invoke CD8s. But what you can also see is that some of these blood stage vaccines have shown very low rates of protection. And one of the things, so you can see all these zeros here, a bunch of these are blood stage vaccines. You know, you have liters of blood and you can have single digit or even double digit percentages of your red blood cells infected. Now, double digit percentage, that's hyperparasitemia. You're way past being helped by a vaccine at that point. But one of the reasons we consider the liver stage to be a bottleneck is that there might only be 10 or 100 infected cells, whereas, you know, if you have 1% of your 6 liters of blood infected, that's an awful lot of parasites to try to intervene with on the basis of antibodies. So I have not been such a fan of antibody vaccines, which are highlighted here in the antibody blood stage vaccines, which is what's highlighted here in the red box. What we have spent a lot of time on for the last 15 years or so is focusing on the liver stage. You can see here on the left, this is a mosquito that's injecting these sporozoites from its salivary glands while it takes a blood meal. The sporozoites find themselves in the intradermal space, and they make their way, some of them, to the bloodstream, where they then, like all roads for the bloodstream, arrive in the liver, and they exit the bloodstream, poke their way through some hepatocytes, and eventually find themselves in a hepatocyte in a vacuole. And over the next 6 days, they multiply like crazy in a process called schizogony to make maybe 20 to 50,000 progeny cells in just a matter of 6 days. And if that process successfully completes, then out the other end comes merozoites. You probably know this term because merozoites are also what comes out of infected red cells. So in effect, these merozoites that come out of the liver, just like the infected red cells later on, each of them are capable of infecting the red cell and starting off the blood stage infection. So if you could prevent this, if you could target these cells by virtue of their either preventing the sporozoite from getting in through antibodies or finding out that an infected cell, like a cancer cell, is a marked cell by virtue of class 1 MHC, you could eradicate it with CD8 T cells. But the bar is very high and the clock is ticking. You only have 6 days to essentially kill all of the infected cells before the infection continues. And so this is a eukaryotic organism. So it's not so easy to decide what the most important antigens to target are. There are thousands, there's 5,000 genes in the malaria genome. And at any one time in its life cycle, there's probably at least 2,000 antigens, 2,000 proteins expressed, since, you know, some of these have a lot of homology to human proteins, but many of them are plenty foreign to be recognized as a foreign antigen by the immune system. And so on the left, we're going to start by talking about vaccines that are based on subunits, meaning we're trying to deduce like 1 or 2 antigens that we can make a vaccine out in the hopes that those are the weak link in the parasite. And if we make a vaccine against them, we'll kill the parasite. And on the other hand, on the right, we'll come later to talking about whole organism vaccines, which by their very name are the whole attenuated organism. They're hard to make, but they elicit a sort of beautifully complex immune response that we could either use as a vaccine or maybe learn from and make a better subunit vaccine from. So sporozoites, we're going to start by talking about the most important antigen on their surface, which is called circumsporozoite protein, because it's all the way around the surface of the sporozoite. And this protein, which we'll abbreviate as CSP, is really the antigen that we know the most about in these malaria parasites. It's relatively immunodominant, meaning sometimes when you put it in with other antigens, it's the only response you get. And it is the antigen that most vaccine makers have focused on, including those that have made the RTSS and R21 vaccines. But CSP alone might not be sufficient to achieve sterile immunity. I often think of antibodies against CSP as sort of like the surgeon's scalpel when trying to cut out a tumor. You can get most of it, but it might not be sufficient to get every last drop. And the leading vaccine that is the most clinically developed is one that you've heard about in the news. It's probably what Leonard was referring to. It's certainly whenever there's stuff in the news about RTSS, my father always calls me and asks me if I'm out of a job. And after I tell you about the next few slides, you'll see why we still have a long way to go. So CSP, which goes by the brand name Mosquirix, is a combination of the Plasmodium falciparum CSP protein and hepatitis B surface antigen complexed in such a way that it self-assembles into a particle that can be used to induce antibodies. So over the last several decades, this kind of CSP particle has been reformulated with different adjuvants in a series of clinical trials. And the vaccine schedule has been formulated and reformulated and finally settled on as 25 micrograms IM 3 times at 1 month intervals with a later booster. And this is the format that's going forward at WHO in the current rollout. This has all the usual features of vaccines that are desirable, pretty good stability. And so this was approved by WHO in 2021 with some conditions about how it would be rolled out and how profit would be distributed, et cetera. One of the remarkable things is that I'm going to show you that the clinical efficacy of this is not nearly as high as other vaccines in the expanded program on immunization. And nonetheless, it really remarkably reduces the liver to blood parasite inoculum. But what this highlights is that unless you can get to zero parasites, the amount of clinical disease that can result from just, let's say, one infected hepatocyte going forward is still pretty significant. One of the great things about working on RTSS, if you're part of the trials partnership, is that everything seems to be published in the New England Journal of Medicine. So there's been a whole series of phase 3 clinical trials published, interim results and final results, et cetera. I'll just highlight a few of those studies, the first of which was published in 2011. So these are studies that were done in 7 different African countries after 14 months of follow-up. And the comparator vaccine was either meningococcus or rabies vaccine, depending on whether we're talking about the infants, the 6 to 12-week-old kids, or 5 to 17-month-old infants. What you can see is in both of these groups, there was at this relatively short interval, for example, in the littlest kids, 56% reduction in clinical malaria cases, which, this could be an important factor in places that have high burden of malaria, where have a lot of kids coming in the door with malaria, and it's putting a pretty big burden on the health system. But these are not 80, 90% reductions. When you look at 4 years, that level of reduction is really eroded. And so dropping to like 32% in the little kids, littlest one, or in the infants, and then the cumulative reduction in malaria cases only being 23% compared to controls. So one of the takeaways from this 4-year study that was published in 2013 was that the efficacy over the whole period, the way the statisticians viewed it, was only 16.8%. And if you compare this to most vaccines that are in the expanded program and immunization, they usually avert 90% of disease. So not great. Really kind of honed in on the idea that RTSS effectiveness wanes over time. And you can see here in a variety of different studies what that looked like. 36% waning to 0% in a study done by Philippe Bejon in Kenya. Another following that going from 43% with 3 doses to basically zero. And some of this was improved with the addition of a booster dose. And the reality might be that this vaccine needs a booster dose even more frequently than that. So if we talk about maybe in the 30 percentile range being the expected efficacy over 4 years, that's maybe as good as it gets for RTSS. So one of the things that the WHO had to grapple with is, how do you view this data? And is this a victory, or is this a vaccine that shouldn't be rolled out? So they recommended that the RTSS vaccine, and it always is indicated which adjuvant we're talking about here, since this is the one that's now been settled on AS01, be provided over 4 doses from kids who are 5 months old. And the data review that was done at WHO in 2020, 2021 looked at the number of events accrued and said that this was going to provide a significant benefit for preventing severe malaria. They looked at a variety of different pooled analyses and made the recommendation to go forward. It's one of the things that happens when the efficacy erodes is that there's not yet enough data to say that there's a mortality benefit here, but that WHO does think that there will be a morbidity benefit, and that could help in areas that have high rates of malaria. So during this year, a lot of the news that's been published in the lay press has been about the rollout of the RTSS vaccine and the investment by Gavi and others to roll that out into malaria endemic countries. So there's one of the limiting factors at the moment is how many doses are available. There's only 18 million doses to be allocated to 12 African countries. That includes those that were participating in the initial pilot studies, and then some regions within the other countries listed here. The idea being that they would be applied to the highest burden areas in those countries to try to shrink the malaria map and shrink the burden of disease. Now, there might be a vaccine with a similar mechanism of efficacy that has a higher and better outcomes using CSP antibodies. And that vaccine was developed at Oxford and is called R21. And it is complex with the matrix M adjuvant. And the reason this vaccine might be more effective is really simply that the valency of CSP as an antigen is higher. In RTSS, you are also in effect getting vaccinated against Hep B surface antigen. And in R21, the CSP antigen is present at a higher dose per particle. And so you're getting more punch from the CSP. And consequently, this vaccine has shown higher rates of efficacy, though also imperfect. This is data from a study that was published last year that led to R21 being approved first in Ghana by the Ghana FDA and now endorsed by the WHO. So the second bullet point is a little bit out of date, but you can see here that compared to the control in this large R21 study, you can see a pretty significant effect size and a dose-dependent effect size, showing that the people who were randomized to R21 had much lower rates of malaria, though not zero. And so it may be that over time, R21 could even overtake RTSS as the most immunogenic of the CSP-inducing vaccines. And it's good to have more than one vaccine in the market. As I mentioned, there's a limited supply of RTSS, and so having R21 may bolster that supply. So there are lots of other subunit vaccines that are experimentally in development. Some of them have been based on adenoviral or chimp adenoviral vectors, MVA vectors. There are many subunit protein in adjuvant vaccines that have been developed. And virtually all of these, including RTSS and R21, target only one or a handful of proteins out of the thousands of antigens that the parasite encodes. So if we go to the other end of the spectrum, the whole organism end of the spectrum, we now sort of lose the ability to say exactly what antigens are being expressed, but we take a very old page out of vaccine development, which is to use the whole live attenuated organism, whether it be viral, bacterial, or in this case, AP-complex and parasite, and use it to elicit an immune response. This is the Plasmodium falciparum sporozoite in an immunofluorescence assay. And what has evolved over the last probably 40 years of research, 50 years of research in malaria, are many different ways to attenuate these sporozoites. So we still don't really know how to grow sporozoites in vitro, though that there are some good advances being made there. So for the moment, you can imagine that the starting material for all these vaccines is insectaries full of very carefully cultivated mosquitoes. And when those mosquitoes' salivary glands are dissected, we can do a couple different things to use them as vaccines. We can either inject the sporozoites unattenuated as wild type parasites, let them go through their normal life cycle, but then give a drug, in this case chloroquine, that will immediately kill the merozoites when they come out of the liver. This allows the full elaboration of all the proteins through the life cycle and then stops the parasite dead in its tracks. This is pretty much an experimental vaccine at this point because we don't really have other vaccines where we give wild unattenuated parasites and then a drug and hope that it all works well. It does work well, but it would be simpler to ensure that the parasite will not come out the other end. And so that's been achieved a couple different ways. First, you can irradiate the parasites, in which case they will go into the liver and they will die usually pretty early. And by dying early, it makes them very safe, but it makes them less immunogenic. So another thing we can do is genetically attenuate the sporozoites, GAS, and depending on the gene that we pick, we can either make them attenuate early, make them very safe, but not so immunogenic, or like we're going to do later in maybe the first quarter of 2024, we're going to be testing a vaccine that is late attenuated by virtue of deletion of certain genes that allow it to go through the liver stage and stop in its tracks just before it would emerge. These are therefore eliciting really complicated immune responses. And because they are sporozoites, even though making a complex response is great, they have some limitations. As I said, they have to be made in mosquitoes. If you're not going to have mosquitoes be your little syringes, which is not what we usually like to do, they have to be cryopreserved, keep them alive, and they have to be injected IV, although I'll tell you some data that is sort of potentially upsetting that. So why do they have to be injected IV? One of the things that has been demonstrated over the last 15 or so years was that even though we like non-IV routes for vaccines, the way those routes were used for other kinds of vaccines, IM, for example, really didn't work in the way that we typically think about IM vaccines. So if you take these sporozoites, dissect them out of a mosquito and formulate them and give them either IV or IM, the only route that was protective in the normal dose volumes that we use for humans was IV. So these are some people in human challenge studies who got 135,000 parasites 4 or 5 times. And then later on, we're subjected to a human challenge where we give wild type parasites deliberately and then follow the patients for a month afterwards. What you can see is that the controls all got infected. The people who got 5 doses of this vaccine were completely protected, and the people who got 4, a little less so. So how are these people being protected? Well, one of the things that's difficult about studying sporozoid vaccines is that a lot of different immune responses are induced, but we can't point to any one immune response necessarily and say that that response is the reason that the patient is protected. If they didn't have that response, they wouldn't be protected. If we knew that, we would turn it into a subunit vaccine. And the reason it's so difficult to understand what's going on in sporozoid vaccines is that sporozoites are professionals at going to the liver. And when they are in the liver, they induce CD8 liver resident memory T cell responses. And these responses are not necessarily revealed by looking at peripheral blood because these T cells live in liver sinusoids. Their job, like other CD8 T cells, is to target infected cells that display antigens on class 1 MHC. And so people who do a lot of flow cytometry, including the people in my lab, have all these phenotype markers they use to try to define what liver resident memory T cells are. But one place they definitely are is they live in the liver and they make granzyme B and interferon. And so, a question for vaccine makers is, how do we make a vaccine that tells the immune response to fill up the liver with this kind of cell? And the best thing that we can come up with is to express the antigen in the liver. We thought about other things like, could we just cause inflammation in the liver, put the antigen somewhere and cause inflammation in the liver? Would that lead the T cells to come running? And some tissue beds seem like that might work. The genital mucosa might work that way, but the liver doesn't seem to work that way. The liver needs the antigen to be displayed in it, and we call that trapping. So I'm going to use that term a lot. Prime and trap means that we prime the immune response somewhere that's not the liver, usually the skin. And then on the second dose, we express the antigen in the liver as a so-called trap to make those prime cells from elsewhere come and take up residence in the liver. And this is what those cells look like. This is some immunofluorescence data from our colleague Bill Heath in Melbourne, Australia. And if you look at these little white boxes in the bottom, these are cells that are marked in a transgenic mouse model as resident memory T cells. And you can see that these cells are essentially patrolling the liver sinusoids. And at the top of the box, there's a yellow box, and this is like a T cell that's just passing by, so to speak. And so I always imagine that liver resident memory T cells are essentially like security guards on cubic blocks of liver. And you need a certain number of these resident memory T cells to be able to patrol your neighborhood, looking out for these parasites. And the question is how to get them to go there, and how many do we need over time to make for a very stable vaccine.
Sean, I don't know about other people, but I had an unstable connection there for a while. And can you go back, sort of repeat the last minute or two?
Oh, sure. Did you see the video?
I didn't. Did other people? Somebody answer? Drew?
I did see it.
Okay.
Yes.
Yes.
Okay, I'm sorry. Maybe it was at my end. I didn't see the last two slides or so.
Okay. Well, if others saw it, Leonard, I'll fill you in later.
Right.
Usually I'm competing with my family members for internet, but not today. The short of it, Leonard, is that in this video that's I think playing over and over here, the white boxes in the bottom, these are these resident memory T cells. You can see how they're slow patrolling the liver. And all the other ones, these other boxes are supposed to show cells that are probably just riding the blood through the liver and are only there for a short time. So the goal of our vaccines is to make these kind of liver resident memory cells. So, and the way we do that, as I mentioned, is to have a vaccine that is a heterologous vaccine that we call prime and trap. And what we're trying to do over time is to develop a really elegant strategy that can achieve this in a vaccine that's simple. But right now, the vaccine that we have is fairly experimental. So the idea is that prime and trap is kind of a hybrid between a subunit vaccine and a whole sporozoid vaccine, WSV here. And the reason it's a hybrid is that we think that you probably can get away with resident memory T cells that target just a few antigens if you can figure out what those are. And so we start by vaccinating our animals, in this case, maybe one day our people, using a gene gun against particular individual antigens, certainly against CSP, but we've also included a few other antigens. The way the gene gun works is you shoot in gold beads coated with plasmid DNA encoding those antigens. And when those are expressed in cells in the dermis, the antigens are recognized as foreign and usually can evoke a pretty good CD8 T cell response. But those T cells, they don't know where to go, right? So on a second dose, a week or a month later, in our most experimental version, what we do is simply inject one dose of sporozoites. You'll recall that earlier versions required 4 or 5 doses of sporozoites. But now if we induce just inject just one dose of sporozoites, those as professionals go to the liver and start to do their thing. And in the process, they recruit these CD8 T cells there. So that's the prime and the trap. If we look in mice that get primed and then trapped, the schedule looks something like this. We give our CSP vaccine at the beginning of the month, and later at day 28, we give our parasites. We call them RAS, radiation attenuated sporozoites. And because we're trying to translate this into something that might be testable in people, we have both fresh RAS, meaning those that were just dissected out of mosquitoes, or cryopreserved RAS, which means they were cryopreserved a long time ago, and they're more like the version that would be suitable for translating this, at least experimentally, into people. And you can imagine that freezing these, even if you intend to keep them alive, might make them a little bit less viable. So this was a question we had to address in this translational project. And then a month later, we challenge them, or we do some kind of immunologic assay to see what kind of immune response we got. So what you can see here is that so first we'll just start with some work that was done by Taylor Olson and Felicia Watson in my lab over quite a number of years. But the takeaway is that in the liver, the highest number of T cells you get that are specific for our antigen and that have the phenotype of being resident memory cells is after prime and trap. If you just give the gene gun or you just give the sporozoites, you get some, but you don't get this kind of 100-fold improvement over that. And the only group that gives you essentially a guaranteed sterile protection is when you give prime and trap. You can see that the sporozoites in and of themselves are actually quite protective. A single dose of sporozoites can protect. This varies, maybe 5 out of 10. In this experiment, it was, sorry, the numbers are hidden behind the Zoom window here, 10 out of 13. But what we never get with irradiated sporozoites, just given once, is 100% of protection. And we really need 100% of protection. We don't get that with the gene gun. Of course, we don't get that in naive mice are all infected. So there was a lot of considerations when we think about, should we take this forward? Could we take this forward? If we couldn't do this with cryopreserved sporozoites, then it wouldn't be a thing. So in order to do that, we asked, could we do this? We knew we could do it with fresh sporozoites. Could we do it with cryopreserved sporozoites? Or could we even do it with heat-killed sporozoites? And what we did there is said, one of the things about a vaccine that is made of sporozoites is that it's alive. It has to get to the liver. If it doesn't get to the liver and do the thing that every sporozoite was born to do, then it probably won't serve as a good vaccine. And what you can see here is that we just took animals that were immunized and then given sporozoites, either fresh, cryopreserved, or even heat killed, and just somebody needs to mute here real quick if you're not. All right. And what you can see here is that the fresh sporozoites, when they go into the liver, there's the most of them in the liver. It's not surprising because they're just right out of mosquitoes. But the cryopreserved sporozoites, about 10 times less of them get into the liver. So we were worried that maybe they wouldn't serve as a very good vaccine. And heat-killed sporozoites, well, they must go somewhere, but they don't go to the places we're looking at in the liver. And so it's been long known that heat-killed sporozoites are not really a viable vaccine, and they certainly were borne out as such here. When we do these immunology endpoints with the cryopreserved sporozoites that don't get into the liver as well, we were pleased to see that they make just as many CD8 T cells in the liver as the fresh sporozoites. And so this is really great. It means that at least the first part of our idea that we could use cryopreserved sporozoites might work. And when we look at the rates of protection here on the far right, in the second red box, you can see that both the fresh and the cryopreserved sporozoites at the doses we were using achieve 100% or close to 100% protection.
Need a dishwasher.
Sally, do you mind muting? I think we're getting some background. So now we've been talking about a vaccine that's experimental and that has to go in IV, but I'm pretty sure that no one here has ever given a approved vaccine intravenously. And so we had a lot of questions about, was there a better way that we could route the vaccine? And this is a story that's still evolving, but I think is a nice combination of what's known about how sporozoites move and how we have assumed that we should inject things in people for vaccines. So these are all the different routes of vaccination that are used in humans, one way or another. Some of them much more common, like IM, subcutaneous, and ID, and IV really reserved for things that are usually like chemotherapy and certain antibiotics, but not really vaccines. Maybe just this experimental vaccine and not much else. So, and there's different kinds of needles, there's different kinds of volumes, there's different kinds of feasibility of injecting in one route or another. But what we do know is that the parasite, when injected by the mosquito, is injected intradermally. And before we had attenuated sporozoid vaccines that could be injected in a needle and syringe, before we knew how to do that, for many decades, you can see papers here in the 70s, this was done by mosquito bite, because mosquitoes are professional flying syringes that we can employ for this use. And when you can take mosquitoes and instead of dissecting the salivary glands and irradiating the parasites, you can just irradiate the parasites, irradiate the whole mosquito cage, and then have them bite people, and that can serve as a vaccine. And when you do that by mosquito bite, essentially exploiting this intradermal route, the vaccines are highly protective. But, and we know that when we do it IV, they are also protected. However, when we do it intradermally, the way humans do it with a needle and syringe, the preponderance of evidence was that this was an ineffective route and that people were not protected. And so why was that the case? This was a work that Felicia Watson did as part of her PhD in my lab. You know, there's all these great reasons to exploit the intradermal route. It is the way the parasites are naturally introduced as sporozoites. The skin is our largest immune organ. It is full of all of these kind of cells here that are poised to recognize and amplify the response to foreign antigen. And it's also known that intradermal immunization can be dose sparing, is relatively pain-free, is safe, and is a route for many licensed vaccines. So if you look at intradermal malaria sporozoite studies, for many years, we have come to the conclusion that intradermal administered sporozoites are inferior to IV sporozoites. You can see here that if you inject parasites intradermally or intravenously, you get more in the liver if you inject them intravenously. And you also consequently get more CD8 T cells. So this was this is evidence that has been telling the field that the intradermal route is inferior. We know that adjuvants can improve the outcome of intradermal sporozoite immunizations. And perhaps most importantly, we have known that the volume is important. And so in our group, we theorized that we might be able to overcome some potentially tolerogenic things that could be happening in the skin with adjuvants. And we might be able to improve the liver burden by paying attention to the inoculation volume. So this is what I mean by inoculation volume. The parasites, these sporozoites, which are a vaccine at one point in their use, and later they're a challenge agent, they move by a motility method called substrate-dependent gliding motility. That means they don't swim through the middle of the pool, they crawl along the edge. They don't have flagella or cilia. They don't swim like Giardia or something like that. And so they can move along the edge, but if they find themselves in the middle of the pool, they will drown. And the middle of the pool means a pretty large volume injection, and pulling themselves along the edge means a pretty small volume injection. So if you look at people who actually study mosquito bites, what they find is that after a mosquito bite, the volume between cells, this sort of interstitial volume, is not very large, but if you inject with an intradermal syringe only 10 microliters, which is smaller than most of you inject when you've ever given an intradermal vaccination, the spacing between cells changes. So we theorized that if we decrease the volume, we could increase the sporozoite liver burden. And there's a lot of challenges to trying to take this forward, but I'm just going to show you some data about how we used low volume injection and an adjuvant to improve a prime and intradermal trap approach. Before I get to the outcomes of the volumes, I'll just introduce the adjuvant that we're working with too. This is a glycolipid adjuvant called 7DW85, and it has what it does is it binds CD1D on NKT cells and provokes an interferon gamma response that can really improve in particular, the development of CD8 T cell responses. So we just recently were awarded with Columbia University a contract from the federal government to work on the translational development of these glycolipid adjuvants, in particular 7DW85. So we've been trying to take that forward with the idea that we need more adjuvants for other kinds of vaccines. And this is a really neat data that emerged from Felicia's thesis, which is that if we take the volume down and at the same time add this adjuvant, we can achieve 100% protection in prime, the gene gun, and trap using intradermal sporozoites. So just to take you through this protection data, without adjuvant, but with an IV route, we get 100% protection. And then if we take our adjuvanted parasites and we put them in a relatively large volume, like the kind you would do if you just weren't thinking and did a regular old intradermal injection, we don't get great protection. And as we de-escalate that volume, we get to a point where we get 100% protection. And that de-escalated volume is just 2.5 microliters, a tiny, refreshing 2.5 microliters. And so what we know is that what we're doing in that approach is that by going down on the volume, we're increasing the liver burden. And that's probably because we're keeping parasites from drowning and they're better able to access the liver. But if we don't have the adjuvant on board at that same ultra low volume, we really don't get protection. So it's this combination of ultra low volume and adjuvant that allows us to exploit this intradermal route and potentially get away from the IV route. I think in the interest of time, I'll just sort of breeze through some of this last few slides. Caroline Duncombe, another graduate student in my lab, has been studying a variety of things, including what the response to the adjuvant looks like. And we can see it, we know a lot about this adjuvant, and even intradermally, it provokes a lot of the desirable responses that would help to drive liver resident memory cell production. But we're going to kind of skip over some of that. I realize we're kind of coming toward the end of the hour. And I think I'll skip over this as well and summarize here to say that it's been neat to be able to show that just because we humans are bigger doesn't mean that the idea that we should use bigger intradermal. Now, granted, all the data I showed you is in mice, but essentially intradermal injections that had been done for many years in mice or in people were done with relatively large volumes. And if we, try to learn from the way the mosquitoes administer, so to speak, the sporozoites, we find that we get to take down the volume, but that alone is not enough. If we take down the volume to 2.5 microliters and add this glycolipid adjuvant, we can have a vaccine in mice that's every bit as efficacious as if we do the intravenous route. So it solves one problem, the ID route, but creates some others. We don't have a device that can do this easily, and we have to consider the use of this adjuvant. But it's kind of an interesting biological premise that is based on how the mosquitoes transmit. What we're trying to do going forward, which is summarized in one slide here, is move away from sporozoites and learn from sporozoites and learn how they elicit the kind of responses they do in the liver. And so as part of another project that we share with colleagues at Johns Hopkins, we are trying to make lipid nanoparticle vaccines that use either DNA or RNA to be targeted specifically to the liver, ideally to be targeted to the liver after oral administration. I was inspired by the idea that a lot of the things we eat go to the liver, and could we use this the exit pathways out of the gut into the liver and exploit them to make a lipid nanoparticle vaccine that would go to the liver and attract liver resident memory T cells. So this is part of a long U01 project that is fraught with lots of technical complexities, but lipid nanoparticles have, of course, been all the rage since COVID, and they can be almost infinitely engineered in different ways using their principal constituents to direct them to go to different places and to be expressed in different cells. So hopefully the next time we run into each other, we'll have more updates on some of those forward-looking nanoparticle-based projects. But in conclusion, I hope that you take away that right now there are 2 WHO approved CSP antibody inducing malaria vaccines. So we can't say there's no approved malaria vaccines anymore. And these are expected to make an impact on morbidity and healthcare demands caused by malaria in parts of sub-Saharan Africa. There are also other vaccines in the pipeline, and sporozoite vaccines are an important part of that pipeline, both for their translational potential, but also because of what they might be able to teach us about how to build even better vaccines. And thirdly, the reality for pre-erythrocytic malaria vaccines is that the T cells need to be in the liver, and making vaccines that induce immune responses in the liver might require that we be somewhat creative, either with heterologous strategies or with kind of fancy new ways of getting the T cells that might be primed somewhere else to go and take up residence in the liver in the hopes that they will elicit a long-standing immune response. Okay, I'm supposed to tell everyone that the word of the day is agent. So there you have it. Hopefully you are paying attention for this slide. And I have lots and lots of people to thank in my group and in the many other groups that we work with, including SCRI and Children's Hospital. And then, we get a lot of funding from the NIH, from the Gates Foundation and others. We're part of the Washington National Primate Research Center, which is really an indispensable tool in trying to develop malaria vaccines because mice lie. And so our monkeys are really important for vetting vaccines. And then I'd be happy to take any questions if there's a little bit of time.
Sean, thanks very much. Unrelated, just I had a fair amount of instability with Zoom. If others did, just let me know. I had a question. Is there value in this disease to the mosquito? What's the whole reason for this? Do they derive a benefit, or they just get a blood meal?
No, so female mosquitoes of all genera need blood meals. The males do not. And mosquitoes that are carrying malaria parasites are generally considered to be less fit than those that do not have the parasites. Now, some of that is probably dependent on how many oocysts they have in their midgut and what that's doing to them. But it's definitely been exploited, or is being exploited by some groups that are trying to develop, transgenic mosquitoes that would be incapable in one way or another of carrying the parasites. So you might imagine that if there were parasites that could not carry Plasmodium and they were metabolically more fit, that they might outcompete parasites in the wild. So that's a whole nother can of worms when it comes to malaria interventions, which is the development of different kinds of mosquitoes and wild releases. It's a hot topic because there's like ethical questions, there's of course lots of entomologic questions, but not thought to be great for the parasite. It's not a mutualistic thing.
When people get natural infection, do they develop protective immunity?
Yeah, so we didn't really even talk about that, but When people get natural infection, usually the number of sporozoites that start that off is minuscule and not enough to be equivalent to our vaccines. So what they really get exposed to is the blood stage. And in highly endemic areas, if you survive and don't die from malaria between ages 0 and 5, usually you develop anti-disease immunity that's based on blood stage antibodies, but usually that anti-disease immunity is not enough to make it complete protection. What we see when we do human challenge in Africa compared to here in Seattle is that in Seattle, when the parasites come out into the blood in people who've never had malaria, it just very predictably has a predictable rise every couple of days with new infections. If you do this in people who have lived in Africa and are living in Africa, what you see is that the number of parasites coming out of the liver is lower, and the slope of the curve of parasite growth is also lower. Sometimes the blood stage immunity can even eradicate that. So it makes it difficult to study as well. But if you don't have that repeated exposure, you probably lose a lot of that anti-disease immunity. And this is one of the reasons that, if we look at the returning travelers, if we look at the people who get malaria in the United States, it is predominantly returning travelers. And the biggest group is what the CDC calls visiting friends and relatives. And sometimes the reason for this is that people who lived in an endemic area, but now have come here and stopped being exposed, have gone back for some time to that endemic area without taking prophylaxis, but they no longer have the baseline anti-disease immunity. And so now they manifest with symptomatic malarial illness.
Not being imported from Africa, but de novo cases in the US.
I didn't hear the first part of your question. I don't know if that's the internet, but I heard you ask if there are de novo cases in the United States. Is that correct?
Yeah, that's correct.
Yeah, so you probably have seen in the news that for 20 years there were no cases. We call those like autochthonous cases. And then just earlier this year, there were a number of cases, first in Florida. And then there was one case, and those were all P. vivax cases. Then there was a case of P. falciparum transmitted in Maryland outside of DC. That also was a local case. And then just on October 3rd, there was a case in, I think it was Arkansas or Alabama, also local case, which was P. vivax. So the TRAPMed meeting was last week in Chicago, and the CDC and the county health authorities from those areas came and summarized those cases. The Florida cases, I think, are the most illustrative. The first patient who developed P. vivax was a person who was at the time unhoused, who had never left the United States. And it was pretty clear to the Florida health team who evaluated the patient that they should take this seriously. So, and they did a lot of spraying consequently, but presumably someone with P. vivax malaria who got it somewhere else had come into that part of Florida, transmitted to local mosquitoes, and then there was limited local transmission. All the cases in Florida were within 4 miles of each other. And, I think the Florida approach was active case detection, and then also a lot of insecticide spraying, et cetera. There were these cases periodically more than 20 years ago, sometimes around the airports in DC and Atlanta. And but I think the fact that there's been, I don't know, 8 or so in this year in 3 different states means that we're going to see this happening from time to time.
You didn't comment on the different types of malaria parasites in developing a vaccine. Is that another complexity, Vivax versus falciparum, et cetera?
Yeah, we would like one vaccine to rule them all, of course, but you can imagine, especially for proteins on the surface of the parasite, there's a there's an incentive to diversify those antigens. So like CSP on P. falciparum, for instance, would certainly not protect against P. vivax. So the hunt is on for conserved antigens that would provide for a pan-malarial vaccine, or a technology that would allow you to pack multiple antigens into one vaccine. But it might be that we have to have regionally deployed vaccines. Of course, Vivax is much more prevalent in Southeast Asia than in Sub-Saharan Africa. Currently, P. falciparum is in the crosshairs because it causes the most death. And then number 2 on the list would be P. vivax. We'll see if the few P. vivax cases in the United States sort of ratchet up the importance of a Vivax vaccine. I don't think so, because it's just a smattering of cases, but we'll see.
Well, unless we have any other questions, thank you very much. Something that allergists, I guess at this point in our profession, don't deal with, but something of worldwide importance to physicians in general. So thank you very much.
Yeah, great. Thank you. Nice meeting you a while back, and have a great day, everyone.